In this video we look at the structure of DNA. So DNA is obviously a really important molecule in biology, and understanding it and the way it works is key to a whole lot of things in this course. So why is DNA so remarkable? Clearly we know that DNA is at the center of genetic inheritance, and that's a key part of evolution and all parts of life. The other thing that's really quite extraordinary is that all living things that we know use DNA in order to reproduce and pass on that genetic information. So all living things have DNA, the same chemical structure and pattern. And so that's a really good evidence that all life on Earth evolved from some sort of single common ancestor, because it's unlikely that DNA could have evolved independently, and so it's much more likely that there was one original ancestor where DNA developed and all life came from that. So before we can really understand DNA, though, we have to go back and understand a little bit of chemistry. So we've got some notes here to have a look at, and just making sure that you have an understanding of all these different things. So firstly, DNA is an organic compound. So organic compounds are complex molecules associated with life which contain carbon. When we look at a chemical structure like this in a minute, all of these corners which don't have a letter there, it's because it's carbon, because we would get so sick of writing carbon all the time in every biomolecule that they're not put on there. So pretty much all of those ones that are joining up are carbon. Carbon's great because it can form 4 different bonds which links lots of things together, and you need that for complex organic molecules. It's a polymer. So polymers are large molecules that are made up by linking together chemically monomers by a process of polymerization. So monomers link together, make a polymer, and that's how you make big molecules. And big molecules are called macromolecules. So there's some terminology that we need to know. There are 4 types of macromolecules that we find in living cells. Nucleic acids. So obviously DNA, deoxyribonucleic acid, is a type of nucleic acid. So is RNA, ribonucleic acid. Proteins, which we talk about later on in the course, and also sugars, carbohydrates, those sort of related molecules, and fats and lipids. So those are the 4 macromolecules that we find in biology. So as it says here, let me just allow that one. That's the S version, which I prefer, rather than the Z version, but it's up to you. So polymers are formed by covalently bonding or joining monomers together, and these are strong bonds that are hard to break. So this is all joined up and connected. But these polymers and other macromolecules, in order to function, they need to make associations or weak bonds with other molecules. So there are other types of bonds where molecules will kind of stick together a little bit temporarily and come apart. So covalent bonds join those parts together. These weak bonds allow them to interact. So some of the things we're talking about there are electrostatic interactions, positive and negative charges, hydrogen bonds, which is what holds together the 2 strands of DNA, as we'll see in a minute, and hydrophilic, hydrophobic interactions. Now this is about molecules or parts of molecules that want to be in contact with water and those that don't. You can click on that link to get some more definition around that, but it'll also be talked about in proteins. So let's have a look at DNA. So DNA is a macromolecule of nucleic acid type. It's made of monomers that are called nucleotides. So and it forms double strand, as we see here. So the nucleotide has a phosphate, a sugar, and what we call a nitrogenous base. Now the phosphate and sugar is the same for every nucleotide, but then there's 4 different types of nitrogenous base. It gives the nucleotides their names: adenine, thymine, guanine, and cytosine, or we usually just call them A, T, C, and G. So here's a nucleotide with the phosphate, sugar, and base, and these are covalently attached to each other on what we call the sugar-phosphate backbone. So phosphate, sugar, with the bases sticking out the side. And there's another strand that's covalently bonded. But these 2 strands together are not covalently bonded together. They bind by these hydrogen bonds, which are weak. That's just another way of showing it. So this is covalently bonded down the sugar-phosphate backbone, but these 2 sides bond with each other, but that can come apart. And that's the really important thing about DNA, is that the information sequence here is preserved because those are connected to each other and stay in order. but the DNA molecule, the information that's in there, can be copied because those parts will come apart during replication, allow a new copy to be made, and then come back together. So the information's stored, but it's also accessible. And this combination of being safely wound up within that double helix, keeping that information safe, but also openable because those weak bonds can be pulled apart. The hydrogen bonds can be pulled apart so the DNA can be read, is really the key thing about DNA and its structure. So on a larger scale, DNA in cells, when we can also see this in the chromosomes page, in prokaryotes, it's an unbound loop of DNA in the nucleoid region. So here's a prokaryotic cell, doesn't have a nucleus, but the DNA tends to be all clustered together in this nucleoid region. If we burst open that bacterium, you can see here's the DNA that's spilled out using scanning electron microscope. This is a transmission electron microscope looking through that cell. There's also some small little loops of DNA like you see here, and that's called a plasmid. So a bacterial cell will have one big chromosome of DNA, but also perhaps some little loops called plasmids. In eukaryotic cells, and you'll have to look at the chromosome page to see that, DNA is in linear chromosomes, which have a start and an end. For humans, we have 46 of them, and they're in the nucleus. But we also have small loops of DNA, a little bit like plasmids, that are found inside the chloroplasts and mitochondria, because they have their own DNA. And if you want to know more about that, look at the endosymbiosis video. So this stuff's covered in chapter 1, and there's also some additional information here about the discovery of the structure of DNA. So here's a very famous diagram in biology. This is Crick's first DNA Doodle, and that's a drawing that Francis Crick did of DNA. Watson and Crick receiving the Nobel Prize for the discovery of that structure and all of the fame, but actually their work was based on the work of Rosalind Franklin, DNA's unsung hero. And this is an X-ray crystallography diagram that she produced that allowed this structure to be determined. So watch that video if you want to know more about Rosalind Franklin.